Hi, this is Dr. L, and I'm going to give you a short lecture on enzymes and metabolism, just the basics. Pause for a moment and read through the list of learning objectives before you continue with this video. What is an enzyme? Enzymes are biological catalysts. A catalyst is a substance that increases the rate of a chemical reaction without itself being altered. Enzymes can be made out of protein or RNA, and we're going to focus on protein enzymes. What enzymes actually do is reduce the energy of activation required for a chemical reaction to occur. That energy of activation is depicted here with this little hump in this free energy diagram. This is the progress of a reaction without enzyme. Over here, the progress of a reaction with enzyme. In both cases, these figures are showing from left to right change over the progress of a chemical reaction. And the vertical axis is the energy associated with the chemicals in this system. So the reactants are the same whether you have an enzyme or do not, and the products are the same whether you have an enzyme or you do not. What's different is the energy of activation, and the energy of activation is the energy that you have to put in to break the chemical bonds in the reactants and allow the formation of something called the transition state, which has to happen before products can be formed. In the presence of enzymes, you will have a reduced energy of activation as compared to the same chemical reaction without enzyme present. So again, it's really important to note that enzymes don't change the free energy of the reactants or the products. They simply reduce the energy of activation inherent to this or any chemical reaction. Now, I said any chemical reaction, but let me correct that. Enzymes can catalyze exothermic reactions, and those are reactions where the free energy of the reactants is greater than that of the products, such as this one that we just looked at. Free energy of the reactants is greater than the free energy of the products. Here we see free energy of the reactants is less than the free energy of the products. That's an endothermic reaction, typically associated with a positive delta G. And this figure is actually incorrect because this figure is suggesting that an enzyme could facilitate or catalyze this reaction, and that's not actually true, and we will get to that in a subsequent video lecture further ahead in this series. So this sort of figure is not true. Okay, if you could take this and redraw it and boost the energy of the reactants and lower the energy of the products, then it would be true, but this is actually not true. So enzymes do not catalyze reactions that are endothermic just all by themselves. Okay, there's a situation called energy coupling where that can occur, and we'll talk about that, but they cannot make a reaction possible if it's not spontaneous. So some terminology around all of this. When you're discussing chemical reactions that are being catalyzed by an enzyme, instead of using the word reactants, we use the word substrates. So substrates are the chemical reactants, and they are going to bind or stick to a particular place on a protein enzyme, and that particular place is called the active site. When they do so, the protein enzyme will switch shape, and it will take on a new shape or conformation. This is called induced fit. It's been induced or turned on by the substrate. In this drawing, the gray is the protein enzyme, and just look at it for a minute and note that its shape is different here, here, and here, and then here, right? The shape is shifting a little bit. So what happens is the protein enzyme has an active site, and it's shown like this, right? And then this green substrate, which is a reactant, will bind to the active site, and then this protein will actually flex or change conformation. And this figure is not doing a great job showing that change, so you could even modify this with a note where you draw a little bit of a shift in this shape, and that will help this chemical reaction occur by reducing activation energy. More on that in a moment. And then once the products are formed, they will be released from the active site of the enzyme. And the enzyme will be able to repeat this, so it won't be used up. It's recycled. So how do enzymes actually reduce the activation energy? Well, they bind reactants at the active site and they change shape, which is to say they have induced fit or conformation. And what this does is it can bring reactants together in very precise ways, facilitating movement to the transition state. And it can also help to overcome repulsion between electrons that come into contact as new chemical bonds form. And so in those two ways, enzymes are able to reduce energy of activation or activation energy. So enzymes, as I said before, they cannot make endothermic reactions happen all by themselves, and that's why this figure is actually wrong, okay? And they also do not change delta G. That's an additional point to note. Now that we've defined enzymes, let's think about organisms, and let's think about the context in which enzymes work. So let's think about the term metabolism. Metabolism is the sum of all biochemical reactions in an organism. There are two basic sets of reactions within the category of metabolism. There can be anabolic reactions and there can be catabolic reactions, and they are connected in a web of connectivity where the reactants of one reaction will often be the products of another, sometimes anabolic, sometimes catabolic. It just depends. So on the left we've got catabolism, and on the right we've got anabolism. Catabolism tends to be energy yielding metabolism. You tend to have a lot of exothermic chemical reactions here. Anabolism tends to be a lot of synthetic metabolism, synthetic reactions. These will tend to be very endothermic or energy requiring. So for example, the synthesis of proteins, lipids, carbohydrates, and nucleic acids, which are the biopolymers, also known as macromolecules of living things, to make those substances requires the input of energy. Those are endothermic reactions. In order to make them, you have to use a chemical called adenosine triphosphate. Adenosine triphosphate is produced as a result of energy yielding metabolism, which is catabolism. And these are strings of exothermic reactions that happen in the cell. So things like all of the chemical reactions that make up what is called aerobic respiration, or in other creatures, anaerobic respiration. Those will produce energy, which gets harvested or conserved, put into ATP, and then the ATP gets fed into these anabolic reactions, allowing reactions that at their heart are endothermic to occur. So there's this web of connectivity. Break things down to reduce energy, catch the energy in the form of ATP, and plug it into reactions that require energy, which is anabolism. So why do we need to have enzymes in order for metabolism to occur? For one thing, we cannot allow the release of a ton of heat in cells. Okay, so this cannot happen, right? So in order to contain that, we will use enzymes, and we will break apart exothermic reactions so they occur in multiple catalyzed small steps, thereby avoiding the release of a lot of heat and allowing the energy to be captured and used to make ATP, something called energy conservation. Secondly, we need enzymes to facilitate endothermic reactions that the cell needs to do by linking them to ATP. More on that later. So but a little bit now, sorry. Endothermic reactions, examples of these are the synthesis of proteins, lipids, carbohydrates, and nucleic acids, or anything to do with transport, whether it's moving chemicals in and out of cells, or whether it's moving whole cells or structures around within a cell, and other things. There are all kinds of reasons that the cell or organism needs to run endothermic reactions. So the way that you get endothermic reactions to occur is called energy coupling. And I will talk more about that in a future video. But basically, in those reactions, you take an endothermic reaction and you find a way to make it exothermic. And generally this involves connecting it to ATP, which is called energy coupling. So what affects how well enzymes do or do not work in organisms or also in test tubes, if you're just studying them? There are many things that affect enzyme activity or function, such as the concentration of reactants or substrates, temperature, pH, or the presence of inhibitors. Here's an image that depicts how substrate concentration can affect enzyme activity. The pink disc-like structures, they kind of look like kidney beans, are enzymes, and the little dished out area is the active site for each enzyme, and the little purple dots are the substrates. So in order for the enzymes to catalyze a reaction involving these purple substrates, the purple substrates have to bind to the active site of the enzymes. And so if you've got a fixed amount of enzyme in a solution, if you add a little bit of substrate concentration, then every little bit of substrate is going to be able to bind to an active site and reaction is going to occur. But because you added only a little bit of substrate, you'll get only a little bit of product. So you'll have fairly low levels of enzyme activity. The reaction rate will be relatively slow. As you keep going, if you increase the concentration of substrate, you will reach a point where there's a matching amount of enzymes and their active sites to the amount of substrate in the environment, and that's where you will see the optimal amount of enzyme activity. Every available enzyme is working. It has substrate bound to its active site at most moments in time. Beyond this, you will plateau when it comes to enzyme activity if you just keep adding more substrate, because adding more substrate will not promote more reactions if you don't also add more enzymes, because the available enzymes will be consumed, all their active sites will be full, and adding more substrate won't make any difference. It won't promote any more activity by the enzyme, any more formation of product by the enzyme. So there's a sweet spot, is what I'm saying, where you're using all of the available enzyme and each or substrate and enzyme, and you're at a point of maximal activity. Below that, you'll see a drop in activity. Above that, you'll see no change. Temperature affects all chemical reactions because the higher the temperature is, the more kinetic energy reactants will have, and this increased kinetic energy will allow for a more rapid movement into the transition state and therefore reactions. So increasing temperature in general will increase all chemical reaction rates, whether they're catalyzed by enzymes or not. However, if there are enzymes involved, there's a sweet spot for activity, or an optimal spot. So if we graph the reaction rate in an enzyme catalyzed reaction versus temperature, what we'll see is if it's too cold, you won't have any activity. And as you increase temperature, activity will increase up to a certain point. And that certain point is called the optimum temperature. This is where the protein enzyme is working best. And this figure here on the right, let me slide that in a bit. This is the protein enzyme, and it's going to be folded and organized to have a certain shape or conformation that is ideal for its function as an enzyme. If it gets too hot, what you will see is like this sudden drop in enzyme activity. And the reason for that is the three-dimensional structure of the protein enzyme is broken by heat. So the chemical bonds holding the enzyme together are busted apart, and the whole thing comes apart, loses its 3D structure. Another way of saying that is it denatures, loses its nature, loses its intrinsic shape. And if it denatures, generally, if it's just a little bit, it can go back, which is renaturing, but pretty quickly you reach a point where that denaturing is permanent and you've lost all functional enzyme. This is just exactly what happens when you crack an egg into a hot pan and the egg proteins immediately denature. They change color, they get stiff, and they don't go back to being, runny egg proteins. Every enzyme also has a pH or acidity optimum, and that curve tends to be a little bit more bell-shaped, wherein there is an optimal pH or acidity at which the enzyme functions best. And beyond that, or below that, you can have loss of activity due to the protein enzyme denaturing, just like with temperature extremes. Or you can also have effects where the acidity affects how the substrate can or cannot bind to the active site. It can change the functionality at the active site region. So there's an optimal pH for all enzymes. Other things also affect enzyme activity. So for example, enzymes can be influenced by chemicals other than the substrate. And when that happens, if it involves an area of the enzyme other than the active site, it's called an allosteric effect, or allostery. So you can have something called allosteric inhibition and something called allosteric activation. Inhibition means the enzyme's not working as well. Activation means the enzyme is working. In fact, maybe it wasn't working before it was activated. Let's do inhibition first. Here in gray is the protein enzyme, and here is the active site. And green is the substrate or reactant that will bind to that active site. At another location on the protein, not the active site, but another location called the allosteric site, it's shaped to allow an inhibitor, which is just some other chemical in the system, to bind. And when that inhibitor binds, it causes the protein enzyme to change conformation, closing the active site and preventing the substrate from binding. So this additional chemical binds to an allosteric site, triggering a shift in the protein shape, which blocks the substrate from binding. And that's very common. It's very common in biochemical metabolisms, biochemical reactions, to see, allosteric inhibition. It's also very common to see allosteric activation, where the protein enzyme has an allosteric site and some other chemical acts not as an inhibitor, but as an activator. And when the activator binds, it makes the active site of the protein enzyme open and be receptive to substrate. So the enzyme can't actually do its job until the activator binds to it, and it triggers opening of the active site and the functionality of the enzyme. So these little inhibitors or activators, these would be extra secondary chemicals or molecules that are controlling the function of the protein, either turning the enzyme off or turning the enzyme on, depending on the context of the particular enzyme. There's a third form of, or not a third, but another way, really, that other chemicals besides the substrates can influence enzyme activity. And this is something called competitive inhibition. And it's competitive because it means there's a chemical other than the substrate competing for the active site. So I took this from the BioNinja site. I forgot to reference it, but you can easily look it up there. So here's the enzyme, and here's the substrate. And normally the substrate would bind in this triangle-shaped active site. And then after that binding occurred, catalysis of the reaction would occur and products would be released. That's like business as usual. In competitive inhibition, what happens is you have some other chemical called the competitive inhibitor, and it is shaped similarly to the natural substrate, so similarly that in fact it will bind to the active site, displacing or blocking the substrate from attaching. And when that happens, the enzyme can't do its job on its regular natural substrate, and so there's no reaction. And by the way, competitive inhibition and also allostery are taken advantage of in the pharmaceutical industry. If you understand an enzyme well, you can often design drugs that will act as inhibitors or activators, allosterically or competitively. And by controlling enzyme activity, you can control outcomes in the body like inflammation or other chemically dependent processes, and therefore manage or treat different forms of disease. So when you're studying enzymes, you have to have a way to study them. And the basic approach is to set up reactions, often in a test tube, and monitor either the formation of the product, which should go up as the enzyme works, right? Or the loss of reactants, which should go down as the enzyme works, reactants also known as substrates. Another way to approach this is to use not the natural reactants and not monitor for the natural products, but what are called fake substrates. So we're going to do that in a lab that we're going to conduct in class as part of class activities. We're going to study the enzyme lactase, and instead of using the sugar lactose, which is the natural substrate for this enzyme, we're going to use the chemical ONPG. So up here on the top, we've got lactose, which is milk sugar, and then just written as a word is the enzyme lactase. And then over here we've got glucose and galactose. So this is the natural chemical reaction. What happens in your small intestines, you take in milk sugar, lactose, and then if you've got active lactase enzyme, it takes the lactose and it breaks it apart. This is a decomposition reaction. It breaks it apart into its substituent monomers, its building blocks, glucose and galactose. And that's great, and it's a really interesting enzyme to study, but it's actually kind of hard to study exactly this chemical reaction because lactose, glucose, and galactose are all clear, colorless sugars, and it turns out to be a little bit hard to measure how much of them is present. So instead of using the natural substrate and looking for the natural products, it's much easier in lab to use a fake substrate, ONPG, which is orthonitrophenyl beta galactoside. And it turns out that this sugar, this fake sugar, is similar enough in shape and size to lactose that it will bind the active site of lactase enzyme, and the lactase enzyme will catalyze the hydrolysis of this compound, and it will break apart and it will release galactose, just like in the normal reaction, but instead of glucose, it will release this chemical called orthonitrophenol, or ONP. And the beauty of that is that ONP is yellow. So this is lactose solution, right? Glucose or galactose would all be clear. ONP containing solution is yellow. The more yellow you have, the more this reaction happened. The less yellow you have, the less this reaction happened. And so by monitoring the formation of this yellow product, we have this great system for studying lactase enzyme. And we can manipulate things like pH or temperature, substrate concentration, and then we can immediately see the effect of those manipulations on the activity of this enzyme. So I've given you a lot of information. Go back and review if you need to. And the next thing that you could move on to is the idea of energy coupling and ATP.